we get experimental science. We're curious, non-judgmental.
It is in fact on frogs. So this study is looking at bioluminescent frogs. These frogs glow in the dark, and they are not necessarily as visible to us, but their wavelength and their vision allow them to see this very, very different world than what we do. And so the fact of bioluminescence, it can happen in many, many different ways and happens in many different creatures. So when an organism absorbs light at one wavelength and emits it at another. So if you absorb wavelength of blue light and emit red, for example. So that's a means by which this happens. There's other animals that bioluminesce beyond the frogs that we're going to talk about today. There's flying squirrels, there's wasps, there's platypi, and more. So discovered in lots of mammals. Maybe we're all bioluminescent. We don't know yet. It could be, Cliff. Yeah, could very well be. So maybe, as Cliff said, many mammals could also be bioluminescing. The first time this kind of biofluorescence was discovered in frogs was actually 2017. And then ever since that discovery, people have been looking for more and more instances in frogs. And so the way they've done this is that it bioluminesces or biofluoresces if you use the right light source. And so what they ended up doing, in the past, there have been two light sources. So that one was violet or ultraviolet that people use to see if there's biofluorescence. So what they did was, these researchers got 528 frogs in Brazil, Colombia, Ecuador, and Peru, and shined 5 different wavelengths of light on them. Everything from greens, reds, infrared, ultraviolet on these frogs to see what lights up. So they're trying a large number of frogs and a large variety of lights to see if, what exactly is a true number. So from 528 individuals, encompassing 151 species, they all lit up, but not with every wavelength of light. Some level of biofluorescence. So that was the catch. That's why this was such a big, impactful study, is that they tested different wavelengths of light on these species, and they did not bioluminesce with every single wavelength. Right, it was with certain wavelengths that they lit up with different lights. So sparky, meaning that they tested 5 different light sources of different wavelengths of color, and some of them lit up with different ones. That makes sense. Sort of like a solar panel, they only glow in the light. So yes, so sparky, the light has to shine on them. So the light energy gets absorbed, and then a different wavelength of light gets reflected, which is the color. So the one that comes in is a certain wavelength of light. The one that's emitted is a different wavelength of light. So the energy that gets absorbed and then reflected, they're not having to produce the light on their own and store up the energy. It's only under a certain wavelength of light. So if they have sunlight coming on them, for example, that's encompassing all wavelengths of light. And then so if you get hit with the right wavelength, you'll emit a different one. Now what it means is you have to have the right vision to see that, which other ones, our eyes might not be able to see it, but if you have it under a certain wavelength, you can. So it's similar way of how fluorescence works in immunohistochemistry, where you do imaging to light up a certain fluorophore, which is when you put on something that glows. You read that it glows, but it doesn't glow to the naked eye. So then when you're shining on a particular light, you excite with a particular wavelength, and then it emits at a different wavelength of color. Reflected or emitted? I would say emitted then, because to me, and this could be difference in physics versus biology, but in the biological context, if it's reflected light, then it's one wavelength of light coming in and the exact same wavelength of light coming out. Here, it's an emission because you have one wavelength of light coming in, and it's a different wavelength of light that's being then emitted. At least that's how I understand it. So sparky, fruit flies don't have this biofluorescence, but they have unique patterns of light that only can be visible to their eyesight, at least we think, in other insects. Meaning, they have these things called wing interference patterns. As their wing moves, you see a prism pattern, and that's actually being reflected. That's not emitted, that's a reflected light. So the wing interference pattern, light comes in and it bounces off, and you actually see a rainbow pattern on their wings. Normally, your eyes cannot see that, Sparky. But a fruit fly eye is designed to see that. And so it's not clear then if a predator would also be able to see these frogs, or if it's a frog-specific thing. Maybe it's a the hypothesis in the paper, it's a way that they can communicate, whether it be mating, predator threats, other things, is a hypothesis that it's driven. Some bounce emitted as an energy. My cells are going to react and make some energy light to send out. I think that's a good way of thinking at aerospace, is that you absorb some of the light and then you emit other wavelengths. And so again, it's that transition between one wavelength of light to another that makes it emitted, because you've absorbed it and now you're emitting a different wavelength, versus like the wing interference patterns that the fruit flies have. That's just straight up light is bouncing off the wing, not being absorbed. The whole spectrum is coming out. So Sparky, these are wing interference patterns. And I like this figure a lot because on the left-hand side is what the fruit fly sees, and that same wing is on the right-hand side under what we see. And so Sparky, you can see, no pun intended, this pattern here, for example, looks very different from what the fly sees. Same thing with this one, same thing with this one, same thing with this one. You and I are seeing drastically different wings than what the flies see, and these contain information in them as well. But yeah, so this is the reflection, and so you can see, Sparky, that we see things differently than the fruit fly would see it. And I think that's super cool, number one, right? That we have these two visual outputs, and if you compare the fruit fly versus what the human sees, and you can imagine this is a kind of principle that governs what happens with the frogs, right? Because if the frogs maybe are seeing things that they can see, but we can't see, right? It could manifest as something like this, where we see maybe, they have some colors, but nothing really, versus they have the vision to be able to pick up on those. And it depends in part visual range. So even this, Sparky, we as humans, to see that right-hand column, cannot see these colors unless you use LED lights, which I think is also kind of gnarly, right? If you use a halogen bulb, you don't see these prism patterns. And so this is one reason why these interference patterns are relatively new in description, because LED bulbs in labs on microscopes are pretty, past 15 years. It's a new thing, and all of a sudden someone's like, this is a whole color range I have never seen before. Back to the frogs. The figures in this paper, the frog paper, are beautiful. I'm very excited to show y'all. So what they found, again, all 151 species had some degree of fluorescence. Sparky, I said all of them, right? It was under different lights, and the intensity was different. Their fluorescence intensity was also different. So they had a very sensitive camera to be able to detect differences between the animal and the background. Meaning that when you're shining these light sources to detect if there's fluorescence, sometimes in the background there might be like a colored hue, right? And so you have to subtract that colored hue from your final measurement, so you know how much is actually from the animal that you're studying versus just background. And so after they did that, they found that some, if you compare it to the original light source, some have about 2% fluorescence, others have over 90% fluorescence. So those are very, very different ranges, but they had the machinery to detect that. They also pointed out in the paper that before this study, there were less than 20 frogs that have been considered to be fluorescent. So imagine, this paper came out in August, at least the preprint dropped. Before this paper, 20 frogs are fluorescent. After this, you add 151, so you're 170 plus all of a sudden fluorescent frogs. Like, think about how game-changing of a paper that could be. I think it's super cool. They found that frogs that were fluorescing either orange or green were most intense under blue light, and their explanation was that similar to twilight is why they were so responsive in that particular color range. Again, as Cliff mentioned, it was thought to be like this fluorescence in land-living animals to be rare, but now it seems like it's everywhere, and it's just you have to look for it in the right way. They were saying in the study, different body parts fluoresce at different intensity. You should have tested it before. But one thing, right, is funding. There's not going to be a lot of money to check glowing frogs, even though there might be long-lasting benefits to it. And then also, like they were saying in the study, they didn't have the appropriate sensors, and other studies had only looked at one wavelength of light. So one thing that they're also demonstrated as a really big region that was fluorescing was right here on the chest of the animal, and that's because that's where they croak. That's where that expands. They primarily make sounds in twilight, which is the same wavelength of light that was really, really intense fluorescing. So if this is expanding, that might make the display of the mating call more noticeable. And so they suggested it might be sexual selection, or it could be something with predation as well. So it's difficult to know which one. So one issue is that they couldn't test function of these. It's a lot of it that is still hypothesis driven for it. They are saying the follow-up for this study is demonstrating if it influences mate choice. So just because they see it in the animal's vocal box doesn't mean that they're necessarily, it connects to there being sexual selection and mate choice. There's like, what is the function of this? Why do these frogs have these biofluorescent markers? Why are they able to do this, and what purpose does it serve? And again, if they're only asking it in the question of sexual selection, of mate choice, that's not the only answer. There must be other functions to this as well, other reasons as to why we might be observing this in nature, and the catch is that it's only going to be visible if you ask the question correctly. If you don't ask the question correctly, you're never going to figure out what exactly the reason is. And so sometimes it's hard, because if you're setting it up in a lab, right, it may or may not be contrived enough where the experiment doesn't work, because you don't have all the ecological factors present there too. Here we go. So these are some examples of what these fluorescing frogs look like. And it again shows you the different species in each of the panels, and it shows you what you can expect. And again, there's a little image here that shows what they look like under the full spectrum light, and then these are the different wavelengths of light that they found max fluorescence in, which I think it's remarkable there's something like this. Even if you see this one's one of my favorites, because the eyes are the things that are fluorescing, and it just looks really spooky. Yeah, it's all you can see is these glowing eyes. And as they mentioned in the study, you see these different patterns, and it's hard to know why these patterns are the way they are. Meaning, is this for sexual selection? Is this for something totally different? We don't know. And so they're testing the ones that have the bigger vocal chambers, and that's where that primarily the fluorescence is located, to ask if it's for some kind of communication or sexual selection that's acting. Now, like we said, that may or may not be the correct answer, but I think it's easier to test for that than like what the eyes are for, because that could be for, again, a broad range of things.